Hi, my name is Kayla Watson. My assigned protein is piezo1 with the mutation of arginine 2456 being replaced by histidine R2456H. And the function of piezo1 is to act as a mechanosensitive ion channel that acts as a receptor. So it senses and reacts to allow our ions to have a sense of touch, and this is achieved by sensing local changes in the fluid pressure and responding to that change by allowing ions to enter through the channel and into the cell membrane to regulate those changes. And this gives us a continuous feeling of how our limbs are positioned in processes like blood flow. Piezo1 is located in the plasma membrane of cells, so epithelial cells, endothelial cells, sensory neurons, and red blood cells. So with epithelial cells, it can help control how cells stick together. An example of this is it may sense urine flow or pressure changes in the kidneys, detect sound vibrations in the inner ear, and with red blood cells, it can influence ion balance and hydration. The piezo1 protein is found in Homo sapiens, animals, and pretty much any animal in the animal kingdom. This piezo1 mutation is linked to dehydrated hereditary stomatitis, which is also known as DHS or xerocytosis, and this is an autosomal dominant disease and is characterized by red blood cell dehydration. This abnormality leads to hemolytic anemia, which then results in feeling fatigue due to loss of oxygen transport, jaundice, which is the yellowing of the skin, and an enlarged spleen. Okay, now I will be discussing the structure of piezo1. So the piezo channels are homotrimeric, which means that there are 3 identical subunits, and this is shown through the 3-blade propeller-shaped structure, which spans the plasma membrane, and it utilizes a cap motion and plug and latch mechanism to gate their ion conducting pathways. So now I'll show you my structure. So here are the 3 blades, yellow, green, and purple, and the orange serves as the ion channel, which is flexible to show the opening and closing, which I'll show you in a little bit. This right here, I've used purple clay to indicate the arginine 2456 position, and this red circle up here is the cap-like structure that accepts ions into the channels. Okay, so as I mentioned, this is the arginine 2456 position, and whenever the histidine mutation occurs, the structure does not really change that much. The only thing is this basically replaces that. So this is the histidine R2456H mutation that occurs. And also I would like to mention that the 3 blades that I mentioned work together to regulate the ion flow. And when there are changes in tension of membranes, the piezo protein essentially just opens up and allows the positively charged ions to go through the cell and then into the cell membrane, which I can't really show on this, but the cell membrane would be like under here. So piezo1 becomes more active after the mutation occurs. So before the mutation, whenever it's just arginine at position 2456, these channels close more. Whenever an ion's trying to come through, they can't get through down into the cell membrane. But the mutation occurs and histidine places that, the channels begin to essentially stay open longer. So they stretch. As you can see, these channels are very flexible, and so whenever it stretches, more ions are just getting through. As a result of this can lead to too much calcium being let in. So the red blood cells are affected because they become more fragile and more likely to be destroyed by the spleen. And the organism is affected because it can lead to hemolytic anemia, which can result in fatigue, jaundice, which is the yellowing of the skin, an increased heart rate and dizziness, and just a decrease in oxygen levels overall. Okay, so the normal amino acid, which is arginine, it is hydrophilic and polar, and it's positively charged. It's one of the largest amino acids as it has a large side chain, which is the guanidinium group. I'm sorry if I pronounced anything wrong. Which has the ability to form up to 6 hydrogen bonds, and arginine can participate in ionic interactions. Since it has a positive charge, it forms ionic bonds with amino acids that have a negative charge, and it can also participate in hydrogen bonding with polar side chains or backbone atoms. The mutated amino acid histidine is also hydrophilic and polar and positively charged, and its main functional group is a imidazole ring, and because of that ring, it has like a compact structure in comparison to the other side chains of amino acids, so it's kind of on the smaller to medium side for size. So while my model does help explain the function and general shape of piezo1, there are some limitations. So the most obvious is that this is not to scale. Piezo1 is actually only nanometers in size. And another one is that pipe cleaners are great because they can bend easily, but they do not fully show the actual dynamic movements of piezo1 and the conformational changes that they go through. I also can't show what's happening at the atomic level, like inside. And the pipe cleaners are not accurately showing the folds of the protein and the overall to the fully functional purpose.